name is Brooke Holmes. I'm from the University of Portland, and today I'll be telling you about my study monitoring Xeric species performance on Ecoroofs using UV visible, near-infrared, and NDVI. This is a plant health chemical study. So I'm showing a lot of different spectra to you in the nanometers from our wavelength from 200 to 1100 nanometers versus absorbance. We're mostly going to focus on this red region and the near-infrared region because you can make a lot of really interesting claims about plant health using those. Our study area is the wonderful University of Portland campus, where we have 2 pilot test green roofs taking place on Christie Hall and Mailing Hall, 2 resident halls here on campus. Figure 1 again shows you all the spectral data for all species planted on either roof, and they are not statistically significant or different from one another, which means there should not be confounding variables causing these green roofs to perform separately from one another. So moving on, this data should not have any confounding variables. Figure 3 shows you the mean spectra for all plants planted in one of 3 different soil substrates we designed for this study. All of these substrates were created by students. Microsol is really heavy in small particulate and porous matter, whereas Brookeosol is largely larger particulate matter and sandy, gravelly soils. Here you can see that Brookeosol has a better chemical health profile for the species planted in it, and it is statistically significant that this is true. Figure 4 and 5 both show you the mean spectra for either the sedum genus species or the deadly lanceolata planted in each soil substrate. We have 5 different sedum genus species that we are testing. They're kind of the poster child for green roofs in the Pacific Northwest and around the world. But we want to look at the biodiversity aspect and their interactions with another Xeric species, because these sedum species can be competitive in high precipitation biomes like our wonderful rainy temperate rainforest. The deadly lanceolata is another senescent during the summer Xeric species and is also native to this area. Here you see a really interesting inverse in this plant health. Figure 4 shows you what you would expect, that in the Brookeosol these sedum species are performing better. However, in Figure 5, it is in Microsol that these deadliest species perform better. This shows you that inverse in when sedum species start to perform better, the performance of other nearby species declines. Figures 6 and 7 also show you a lot of spectra for all sedum species and then all of the deadliest species planted with a sedum species. Here you see a really interesting result in that this sedum album, this blue line if you can see it, is not nearly as efficient in chemical analysis as you would expect it to be, as it is the largest growing sedum that we've seen over the 18 months that this green roof has been implemented. Here you see that it is not as chemically healthy, showing you that when sedum species, or really any species of plant, grows larger, their concentration of chloroplast pigment per tissue can decline. This shows you some interesting results in that aspect, because we were not expecting the sedum album species to be anywhere further than the top. Again, this deadliest species profile shows you in another way how when the chemical profile of a sedum species improves or it is growing better, that it will tend to be, that the deadliest species will tend to not survive as well. Thank you.